ORDA Science Webinar, Reference Intervals, An Approach for Quantifying Injury, organized by KCB Snyder and recorded by her via Microsoft Teams on February 25, 2025 at 1902 UTC. Official logo of USGS, Science for a Changing World. Official logo of ORDA, U.S. Department of the Interior.
So, we have a USGS disclaimer you can quickly skim.
Any use of trade firm or product names is for descriptive purposes only and does not imply endorsement by the U.S. government.
But then who are we and why should you listen to us? I'm Richie Erickson. I'm a research ecologist with the USGS in La Crosse, Wisconsin. Primarily, I do invasive species modeling, so big head carp, environmental DNA of carp, and other related projects. But why am I here talking to ORDA? Well, my PhD is in environmental toxicology, and I have a math minor with that, so I've done a lot of modeling and statistics related to ecotoxicology. Also, I do statistical consulting and review at my center, so if people need help with their study proposals or reviewing them, I help out there. I'm also an editorial board member for environmental toxicology and chemistry, so I'm still involved with toxicology and chemistry. And I received a Presidential Early Career Award earlier this year as well. So that's sort of my background, and Barnett can introduce himself as well.
Slide shows a photo of Richie holding skis at an international skiing event.
Hi, I'm Barnett Ratner. I'm a research physiologist, but my entire career in the federal service, 47 years, has really been in the area of ecotoxicology. I do controlled exposure studies, field work, risk assessments, work in the area of alternative toxicological test methods. I'm a consultant to the U.S. Fish and Wildlife Service on candidate materials that take the place of lead used in hunting. And I've done a lot of editorial work for the journal Environmental Toxicology and Chemistry over the course of my career.
Slide shows a headshot photo of Barnett smiling in front of a body of water. Screenshot a paper titled, Moving Beyond P, Less than 0.05 in Ecotoxicology, a guide for practitioners written by the presenters.
Barnett and I were happy to speak to this group. We spoke before. I think Barnett spoke a couple of times. Barnett and I spoke about more statistical work. He and I, through ET&C, were noticing that a lot of ecotoxicology studies had similar pitfalls and some statistical problems. So we got together and wrote a critical perspective to talk about different statistical methods. And this work's grown out of that work as well. And at ORDA, we know statistics are important, and this group collectively, and most individuals probably know more about it than Barnett and I. But just to help frame it for somebody who might not be familiar, there's a pre-assessment and then the assessment and the restoration, and all of these require showing what's going on and quantifying things. So, for example, with the NRDAR process, there's the damage assessment part of it, where you want to show that something bad has happened because of these different insults to the system.
Damages. A legal term for the amount of money sought in a claim made by trustee plaintiffs to PRP defendants. It includes the cost of assessing injuries as well as the cost of restoration implementation.
And then a damage. So this is just a definition of it that you can skim or read. You're probably much more familiar with than us, but just something to point out in here is there's the injury part that shows something bad happened, then also a baseline condition. And we just want to highlight the baseline condition because it's important, because that's what we're going to talk about, how to show what is the baseline condition today.
Injuries. Under OPA, injury is defined as an observable or measurable adverse change in natural resource or impairment to a natural resource service. Injuries can occur directly or indirectly.
And then injury, just a definition here, it's just the measurable adverse change. But how do we show what is an adverse change or an impairment with these different endpoints? It could be something like survival, growth, health, physiology, or these things. So these definitions talk about it, but they don't say what is that difference. If you remember from kindergarten, Sesame Street, one of these things is different than the other. In this case, Bert can see that one of his pigeons is different than the other. But how do we show that, kestrels or pigeons are different than others? Ernest Rutherford, the famous nuclear physicist chemist who discovered the proton, or the nucleus of the atom, said that if you need statistics, design a better experiment. However, that was in the early 1900s, and statistics are now vital to modern science.
Statistics plays a vital role in today's society by providing the foundation for sound decisions. A quote by Robert A. Donnelly in The Complete Idiot's Guide to Statistics.
This is a quote Barnett loves that just talks about how we use statistics for our sound decision making. And science has changed, so we need statistics. But what should we use and how should we do it? And more specifically for this group, how can we show that adverse change happened and evaluate it? So when I've talked to people in this group before, the lawyers always want to show that something's statistically and biologically significant. So what are tools to help with this?
Screenshot of a paper titled Moving Beyond P Less Than 0.05 in Ecotoxicology, a guide for practitioners written by the presenters, and a screenshot of a paper titled Use of Blood Clotting Assays to Assess Potential Anticoagulant Rodenticide Exposure and Effects in Free-Ranging Birds of Prey, written by a collective of researchers, including Barnett A. Ratner.
Barnett and I, again, just talk about some tools in this paper, but we keep growing and expanding our toolbox. I love working with Barnett. I've known him since grad school, almost 20 years ago, and he keeps learning new things. So he and I learn new things together. And out of necessity, we had to learn about reference intervals. Barnett?
Yes. So in 2015, I was approached by a colleague who worked for the Environment and Climate Change Canada, John Elliott. And he was involved in a study, and it was certainly not a natural resource damage assessment. We, and specifically John and Sophie Hinmarch, had collected samples of raptors that had been admitted to a rehabilitation facility in Canada. And they had drawn blood samples from the raptors because they are and continue to be very interested in anticoagulant rodenticide exposure. Anticoagulant rodenticides prolong blood clotting time. And the reason they contacted me, they actually had another investigator involved in their study who had to drop out of the study, and they needed essentially a relief hitter or a relief analyst to analyze a lot of the blood samples that had been collected for blood clotting time. And when they described to me what they had done, my question to them is, boy, how do you analyze these data? What are you using for a control or for a reference? So on the next slide.
Screenshot of a paper titled ASVCP Reference Interval Guidelines, Determination of De Novo Reference Intervals in Veterinary Species and Other Related Topics.
When we finished our analyses, I tried to help them a bit with analyzing what we had found. And I was familiar with the concept of reference intervals that had been used in human medicine and veterinary medicine for decades. Richie, jump to the next slide.
Graph of normal/Gaussian distribution.
So what are reference intervals? They're the range of values deemed to be normal in 95% of a healthy, disease-free population. Sometimes that can really be easy to define for humans and even domestic animals, but very difficult to define for wild animals, fish and wildlife for that matter. So what is the use or application of these reference intervals? It's really to identify clinical features not present in a healthy population, and it can provide evidence that an organism is unhealthy or in a disease state or near or approaching a disease state. Let's jump to the next slide. So here's an example from a human clinical setting. I'm sure all of us have gone for some type of health check for the federal government or just for your own concern. Blood is drawn, and a comparative metabolic panel is often analyzed from that blood sample. And the values derived from that blood sample are compared to a reference range, which is really more appropriately referred to as a reference interval. And for this particular subject, if one looks at the middle of the slide, one sees glucose, blood glucose, which normally falls in the range of 70 to 100 milligrams per deciliter. But this particular sample from the patient had a value of 106, marked as high outside of that reference interval. Well, what's the interpretation? The concentration of glucose is outside the range of a healthy reference population, and perhaps further or repeat testing is needed to rule out diabetes. Let's go on to the next slide. So there are a lot of things to consider when establishing reference intervals. One size does not fit all. One has to consider the species or even the strain or breed. Age, sex, nutritional plane, and reproductive state can affect the measurements. Individuals should be healthy, and that reference population is sometimes very difficult to define what is healthy. As a rule of thumb, 120 individuals that are healthy are used to establish this reference interval for various analyte measurements, but one can actually establish reference intervals with far fewer individuals, 30 or 40. Reference intervals are generally laboratory specific. The next little comment I have, does an abnormal result above or below this reference interval equate to harm or damage? No, that is not the case. Sometimes it could be a low-level subclinical effect that is not really pathologic. From a statistical standpoint, when one runs numerous tests on the same individual, the chances of getting one abnormal result increases for the number of analytes looked at. So for 21 tests, there's a 0.66 probability that one might get a value outside this reference interval range.
P equals 1 minus 0.95 N.
Let's go to the next slide, Richie. So back to this work that was done by Sophie Hinmarsh and John Elliott that I helped with. They had collected blood samples from different species of birds that were admitted to a rehabilitation facility in Canada. They had samples from many species of raptors. This slide shows their data for barred owl, great horned owl, barn owl, and barn owl fledglings. And if one just looks at the spread of the blood clotting time, prothrombin time, for each of those different species, the data are really clumped together for the most part, but every once in a while one sees a very high value, which could possibly be related to exposure to anticoagulant rodenticides. So how did we end up picking out these high values or outliers? We sort of did a no-no. We had all kinds of data from captive screech owls, a healthy population at the research center I worked at. And we established reference intervals for those screech owls. And the solid line between the two dotted lines was actually the upper reference limit. And we even multiplied that value by 1.5, and the very dark line is 1.5 times the upper reference limit. And if one looks at these data points, there are actually over 100, because they're points on top of one another. There are about 8 points that are elevated above the upper reference limit. Let's go to the next slide, Richie.
Screenshot of a paper titled Brodifa cum isomer formulations with potentially lower risk to non-target wildlife. by a collective of researchers including Barnett and Ritchie.
So this was an interesting learning experience, and we have actually used it, Ritchie and I, since then, looking at different isomer formulations of rodenticides in non-target wildlife. Ritchie, let's go to the next slide. So what we have done in our controlled studies, in some circumstances clearly much different than a natural resource damage assessment, we have captive American kestrels. And when we run our studies, long before doing the study, we draw a blood sample from the kestrel to measure clotting time. And this is essentially a reference value. We then, after a couple of weeks, expose the birds to diet containing rodenticide formulations, in this case, rodenticide formulation A or rodenticide formulation B. And after several days of exposure, we draw yet another blood sample. Let's go to the next slide, Ritchie. What we did with the first baseline reference sample was to create a reference interval. There were 27 reference samples ranging from about 11.6 to 15.2 seconds for prothrombin time. We have a mean, a median, a standard deviation, and then we actually end up with a upper reference limit and a lower reference limit. The lower reference limit is 11.79 seconds. The upper reference limit is about 15.24 seconds. Let's jump to the next slide.
Bar graphs showing that birds exposed to the highest concentration of formulation B, 0.50 micrograms per gram, had significantly higher prothrombin time than those exposed to any concentration of formulation A and all lower concentrations of formulation B.
Well, this is what happened after we did our exposure to these different formulations A and B at different concentrations or dose levels for 7 days. And clearly the bar on the right side of the graph that you're looking at, you can see the 4 data points for clotting time, and they are much, much higher than the values in the bar on the left side that has a 0 under it, an unexposed control group. And there's actually a hatch line in the y-axis. So you're comparing values that are about 15 seconds to values at this high dose of formulation B that are well over 100 seconds. So one could do, and we did, standard parametric statistical analyses, analysis of variance followed by a multiple comparison procedure, and clearly the birds exposed to the high dose of formulation B were significantly different in clotting time, prothrombin time, than all the other groups. But another approach to looking at these data is to compare them to the upper reference limit of the reference interval. And that is the dotted line that one sees about at 15 seconds. And clearly, the birds that were exposed to the high dose of formulation B on the right side, those values are greatly above the upper reference limit. The other values are not, except for one, and that is formulation A at a dose of 0.5. That's the bar right in the middle. So although as a group that did not differ significantly from the other groups, there was one individual that had a slightly elevated value, and that would be categorized as a respondent. Let's go to the next slide. Ritchie, at this point, you take over.
Thanks. So Barnett and I worked together on this paper, and we thought the method would also be informative to this group. So for the rest of the talk, we're going to talk about just how to use establish reference intervals to use. So just the first important concept we're going to drill home is it's important to have a good reference population. Then we'll talk through the conceptual steps for estimating the reference interval and the lab or field studies you need to do. Next, just how to do this in R. And then last, just some major pitfalls, because Barnett and I have saw some misuses of this like any procedure as well. So just to drill home this point, hopefully this group is familiar with or like a reference site. So if you're doing a NRDAR project or something, you have your polluted site, which is a Superfund or whatever damage site it is. But then also, how do you show there's something bad? You have a reference site. So it's the same idea where you have a reference population or a reference site. Also, something else, it was drilled into me at CTAC meetings by Wayne Landis is we have references, not control. But if it helps you, think of like, what's your control population? What's your unaffected reference population?
Screenshot of a paper titled ASVCP Reference Interval Guidelines, Determination of De Novo Reference Intervals in Veterinary Species and Other Related Topics from the Journal of Veterinary Clinical Pathology. Tables discussed in this talk can be referenced in this research paper.
And we're going to walk through the steps presented in this paper. I would highly encourage you to read this paper. If you need access to it or can't find it, please reach out to Barnett or I, and we can send you a copy. We're sort of like the movie, but if you read the book, it has a lot more details. So I encourage you, if you want to dive in more, to check out this paper. The book has a table that I'm going to walk through because it's the steps. So let's say you want to show there is an injury or at a damage site. How do you do that? So step one is do a literature search for the information about the analyte to be analyzed. So maybe, Barnett's looking at rodenticide, so it's clotting time with an assay. Maybe it's another biomarker or another important endpoint. Then define what is your reference population. So what is the healthy population you're comparing it to? And then how do you include individuals in this population? There's another table that talks through some of these details, but basically, what is something tenable to say that you're different than? This is a step for the human health process where it's developing the clinician criteria, but something else here, especially for the ORDA group, would be something that the biologist with ORDA and the legal team is comfortable defending. So what is a legally tenable endpoint? And then figure out how many individuals you have or need. So this is like a statistical power. So it's how many individuals do you need based upon your variability of the endpoint. And this might require some iterative studies. So if it's a totally new species in a new system, you might have to start off and just say, take samples to figure out what is an approximate mean and variation. So you can start to crank down on these things. Then select the reference interval. Select the reference individuals. So especially with human or veterinarian health, we look at individuals. I know natural resources is more populations, but it's still important to think, how are you picking your individuals? Next, collect and handle your references in a standard manner. Basically use solid lab and chemistry studies and approaches. Next is analyze the reference samples using established methods. So if Barnett and I were doing this, we'd have to tag team and get a chemist to help us. The part I love as a statistician is now you get to start to look at the data and look at a histogram. And then figure out what are outliers. So what needs to be changed or done to make sure you don't have outliers in your reference population. Maybe you have, like you missed an individual. If it was a human, maybe they had the flu that day or a cold. So they're really not healthy, but they appear healthy. And then determine if you have non-parametric or normal data so you can use the appropriate methods. Next, 11 is calculating the limits. And then I'll go through this table as well just to talk about some of the considerations. Still more steps. So determine if you need to break individuals down. So for example, human health, male versus female, or are you comparing Olympians to couch potatoes? What sort of thing is going on here? Make sure it's a valid comparison. Are you looking at one Superfund site to another Superfund site, or is it a pristine wilderness? Is it saltwater versus freshwater? Just think about how you break things down. And then last is document everything.
Screenshot of an EPAECO update.
So, and again, I'm going to just bring up reference site again. This is a well-known idea for ORDA, and it's the same thing for populations. So as you have to pick a smart reference site for a natural resource damage assessment. You also have to pick a smart reference population for this approach. And conceptually, you can substitute the two ideas and just think about what's a good site. And as a statistician, I don't know the answer. Barnett, as the toxicologist, might be able to help, but it also requires the toxicologist and site biologist and chemist to say, what is a tenable comparison site?
Going forward, Richie briefly references tables full of information that are shown on the screen. To reference the full table shown, please see the research paper mentioned earlier from the Journal of Veterinary Clinical Pathology.
So, as we're including individuals for veterinarian health, it's things like the age and sex, breed, strain, all these sorts of characteristics that change individuals. Clinical, is it somebody who's healthy or are they unhealthy? I know Barnett does procedures with those kestrels to make sure they're in similar health. Location, though, as well. What's a good match? What's not? And then these other things as well. Are they stressed individuals? Are they fasting, non-fasting? These are all experimental design criteria that hopefully you think about anyways, but they still are important here as well. Just another table of things to look at in greater detail are just like, how do you prepare individuals? How do you collect them? What's the handling and the collection and chemistry, analyte stability? So reference intervals aren't magic. You can add them to your existing methods, but all the things people oftentimes consider important are still important. So keep thinking about those ideas. Next is what method do you use? So the reference intervals, they're based upon the distribution of individuals. So do you use parametric or non-parametric? Parametric assumes you have a normal or Gaussian curve. Non-parametric means there's different bootstrapping methods. So you can read through the table on your own, but this is just something to think about, is how many individuals do you have? And Barnett and I also know that it's hard to get biological samples. Human health, they oftentimes can get thousands or more samples to do these things. Veterinarian health, maybe more, but not necessarily. So next, how do you actually do this once you get the data? So this is the fun part for me. For a lot of you, it's probably dragging it across the finish line. So luckily, there's an R package that does this. If you know how you install R packages, you can install it from CRAN and then load it with the library function.
Referenced R packages Finnegan D, open bracket, 2024, close bracket, period, reference intervals, colon, reference space intervals, period, R package, version 1.3.1, comma, less than sign, https://cran.r-project.org/package equals reference intervals. Found at library, open bracket, reference intervals, close bracket.
And then after you get the data in,
histogram showing a rough bell curve relationship between the baseline prothrombin time between 12 and 15 seconds and frequency between 0 and 7.
And also, I'm skipping several steps. I'm not going through how to clean up data or other things. I'm just showing the key points. If you want to walk through the code, follow up with me, and I'm happy to follow up later with a one-on-one. So this is Barnett's 27 individuals, and they appear normal enough. I say normal enough because I personally prefer to look at histograms and the data themselves rather than use formal testing, because formal tests for normality are both too liberal and too conservative. If you have a large enough sample size, the test will tell you that your data is oftentimes not normal, simply because the power of the test increases as individuals increase. So small but non-important deviations from normal appear. Conversely, if you have a very small sample size, you oftentimes don't have the power to detect non-normal distributions as well. So that's why I prefer just to look at the histogram as well. Looking directly at the data is something you should do regardless, because you can sometimes find other mistakes as well. Maybe somebody entered in 25 rather than 2 or something, or 200 rather than 20. So anyways, I just like to look at the histogram to see what's going on with the data, and Barnett's individuals for the reference population seem normal enough. So now that we have the data, we can use the ref limit function. So take the data, pass it to the ref limit function from this package. There's different methods to detect the outlier. We're using the horn method, and then we tell it what are the reference limit and the confidence. And then it bootstraps or resamples 5,000 times within the program. So it's basically like shuffling a deck 5,000 times or rolling a dice 5,000 times to get the distribution. That's important because you'll sometimes get very small differences, like the last digit, just due to random sampling. But then it kicks this out. So the summary tells us, what data did we use? It tells us there were 27 individuals. It says we used a 95% reference interval, and around this reference interval, we had a 90% confidence interval. This can be confusing. So a way to think about this is, conceptually, this is very similar to estimating a standard deviation and then a standard deviation around the standard deviation. So it's an estimate of an uncertainty around an uncertainty estimate, which is a tongue twister, but it's how much variability exists in that range estimate. So next part of the output just says what outlier method you used. No outliers were detected. The reference interval is what Barnett talked about, and then these are the upper and lower limits. So that's the R output.
Line graphs showing parameter per unit, helping to visualize where confidence intervals lie compared to the reference interval.
And then here's a figure. It's not super revolutionary, but it's just a default output that you can plot from the model just to let you see what's going on. So you can see what's the reference interval, and then what's the uncertainty around those ends as well. And that's the case where it depends what you want to do, but if you start to fall in that uncertainty range, it's very different than like Barnett's extreme outliers that were very far away. But this lets you see what is normal or typical. Or as Bert or as Big Bird might say, one of these is different from the other. So reference intervals, what problems have we sought and how do we avoid them? Again, I would say pick a good reference value. Barnett and I have sought where people were using values from, I think, zoos, but the problem was that the database wasn't being used correctly, and the people were accidentally including unhealthy and sick individuals. So they were trying to look at an endpoint, but rather than include healthy individuals only, they also had sick. So the reference values weren't good. So if you're using an existing database, make sure you understand your data, but also choose the appropriate reference values. So like any approach, and even this traditional ecotoxicology, pick a good reference population. And this is a well-known problem, though, in ecotoxicology. I just threw up several classic articles about this that I saw. You know, Peter Chapman's late and great talks about this with his ecotoxicology paper from over 20 years ago now. And a lot of these are older papers. But it's just a big challenge is, what is a relative control population? We can't have control populations, so we have references. So what is an appropriate reference population? And that's something, as a statistician, I can't help you. Barnett, as a toxicologist, might be able to, but it's something for the biologist to think, and also possibly the lawyer, what is a tenable comparison when they're working on the settlement or other parts? So that's the end of our talk, and we have plenty of time for questions. So there might be something in the comments, but I'll, open the floor for questions.
Richie Erickson, statistician. R. Erickson at usgs.gov. Barnett Ratner, ecotoxicologist. B. Ratner at usgs.gov.
There was a comment from, I think, Sarah, if I'm reading her name correctly. In natural resource damage assessment, we look for background levels, parentheses, or levels of a contaminant, but for the release of hazardous substance. In other words, ambient contamination from daily human activities that don't include the spill release. Thank you for sharing that, and that's exactly what you want for your reference population. Chris.
Hi, Richie. When I've tried to introduce the concept of reference intervals to other statisticians, they have pushed back on me and they said they would rather do tolerance intervals. Have you run into this kind of debate, and what's your perspective, if so?
I have not run into this debate, and I'm not familiar with tolerance intervals. So, Barnett, can you comment?
Yeah, Richie, and maybe turn your mic off. And I understand what you're saying, Chris. And as we sort of pointed out in the talk, just because one exceeds a reference interval value, that does not automatically indicate there is damage. And the descriptor that you used is actually an indicator that there is a higher probability that there could be damage. In a sense, it's a health assessment benchmark. It's a somewhat higher value when that value is exceeded, perhaps. It has a much greater likelihood of there being an insult that is judged as being toxic. And it is interesting. I've looked at data from veterinary diagnostic laboratories, and I've looked particularly at data for lead. And lead has a reference interval or range. It's a very low value in tissue, less than 3 micrograms per gram. And although lead has no biological function that is good, there are levels of lead probably in all of us and in many, many species of animals that are low levels, that are not linked to adverse effect. Now, this lead reference interval that I have, the upper limit is about 3 micrograms per gram dry weight. And if one were to have a value of 5 micrograms per gram dry weight, well, that exceeded the reference interval, but it might be not at a threshold that causes an adverse effect. Lead's kind of funny because the adverse effect level over time has gotten lower and lower. But I think what you point out is absolutely correct, that exceeding a reference interval, the upper limit, does not necessarily equate to toxicity. But it does show that it's outside what is considered normal. I hope that answers your question.
I think I realized there are actually two meanings of the phrase tolerance interval. And I think the one you're referring to it comes from a concept called a tolerance distribution, which you establish like a dose response relationship. But there's also something called a statistical tolerance interval, which, so in statistical inference, there's the 3 intervals that people frequently present. Those are the confidence interval, the prediction interval, and the tolerance interval. And that's kind of a purely statistical concept. The idea of a statistical tolerance interval is that you're constructing an interval to capture a certain fraction of the population. And that's kind of how the reference interval is defined oftentimes. But the tolerance interval is constructed in a much more formal way, using all the machinery of statistical inference. There's a book, I believe it's by Hahn and Meeker, called Statistical Intervals, and it defines and talks about those 3 different intervals, and it does not talk about reference intervals. But and I have another book on my bookshelf, Statistical Methods for Medical Research by Altman, and he talks about reference intervals and not tolerance intervals. So it seems like there are different parts of the statistics community that are not talking to each other, and I've always been interested to get people's perspectives on this disconnect. Anyway, thank you.
Thank you.
John.
Yes, thank you both for this presentation. You did a very good job explaining the material in a way that I think most everyone can understand. Maybe just a note, some commentary for folks wondering if this approach has been used in any NRDA cases in the past that are now settled. The answer is yes. There was a few sites in southwestern U.S. where Mike Hooper, who's now retired, and I, and when I was in graduate school, we used reference intervals in a laboratory mallard population, looking at the effect of some acid mine water effects on clinical chemistry profiles for those animals, as well as hematology. That was published after that those cases had been settled, and then maybe not surprisingly, with Deepwater Horizon, one of the co-authors of the veterinary paper for which is the basis for this work presented today, or at least the methodology that Richie went through in detail. Kendall Haar was co-author on, there was at least one paper, I can't remember which species, if it was laughing gulls or cormorants, but some of the Deepwater Horizon related work also used reference intervals as part of what I think is an important point I'm about to say is as part of a weight of evidence approach. So I think those of you who have been around the block a few times know that this sort of approach, I think, would arguably be useful in a weight of evidence approach. There's still the interpretation part that has to be done, right? Which is why Richie was kind of pointing to Barnett, is you need a toxicologist to be able to interpret this and put it in context with what it means for your case, your injury assessment, and any potential associated damages. But thank you both for presenting this. I think it's an interesting and kind of new perspective and way of teasing apart your data and looking at trends and potentially being part of what could be part of a compelling story.
Thank you, John, for sharing your insight. There's a question in the chat from Wendy. Can you use baseline data from the population that is subsequently affected and remeasured for post-disturbance conditions as your baseline reference population? A question mark. How would you address an argument that background, broad-scale environmental changes could be causing the ascribed changes in contrast to assert source of injury? Would you need to have a unique population to use as your reference to avoid this issue? So with Barnett, I'll take a stab at this and then pass it off to Barnett. Statistically, you can crank the numbers. That's not a problem, but it's so like what Barnett did, he exposed the kestrels and then he had a reference populate. He established the reference interval with the baseline conditions, exposed the population, but also had a control in his lab study. So they could say that the control didn't change through time. In this case, if you're using your baseline data, you have to be able to make a convincing argument that only the contamination changed and not something else. For example, maybe it's pollution later, but maybe it was a hurricane or something hit the site. So you have to be able to clearly make an argument that that's a compelling reference population. Barnett, can you chime in more?
Yes, and Wendy, my answer would be yes. And when I say yes, I'm laughing because you either have to replicate in time and space, and I like to replicate in both if you can. So you could have reference sites sampled at multiple times, looking at a baseline condition and then a baseline condition at a different time. And then one could have a baseline condition at a site that is subsequently disturbed in some way, and also sort of simultaneously do that type of analysis and comparison. And I'm a big advocate of having more than one reference site because I have been surprised over the course of my career many times with what we thought was a reference site, which ended up being a very poor choice. I hope that addresses part of your question. Thank you.
You're welcome, Wendy. John?
I'm back. Barnett, the John Elliott and other, I know Elliott wasn't the first author with where they used the screech owls for the reference interval population. But none of the exposed individuals were screech owls. So can you comment on, like you said there was a no-no, but I missed the elaboration part.
Yeah,
Is that
well,
the
that's
no-no of,
owl species can be very, very different. And that was the very best we could do at that point in time. And I actually, since I did the analyses on the samples from the species of owls that were admitted to the rehabilitation facility, I had also done the analyses in our captive screech owls. And there are databases of information, for example, the International Species Information System, ISIS, that essentially lists all kinds of data, a lot of clinical chemistry for hundreds or thousands of species. And sometimes people actually draw from those databases to use the information as a guideline to make decisions. I don't think that's the greatest thing to do, but in the case of the screech owl study and the other owl species, that was what we did was I felt one step better. The analyses were all done at the same laboratory using the same methodology. And although screech owl and barred owl are very different species, it kind of gave us some information on the spread of the data we should expect. And that's why the second study I described with the American kestrels, here we generated a reference interval in an unexposed healthy population, and then ran our test using that same species. And I felt that that type of comparison and the decisions and interpretations we made from that study were far more powerful. So
Slideshow goes back to the bar graph showing prothrombin time of American kestrels after being exposed to various concentrations of 2 rodenticides.
What John Elliott and Sophie Hinmarsh and I did was a little bit of the best shot we could do, and we pointed out the shortcomings and uncertainties associated with it. And then what I later described that Richie and I and other colleagues did was certainly a much better approach. So I hope that helps a little bit with that question and my comment to it.
yes, certainly. Thank you for that, because I was definitely wondering.
Well, I don't know, maybe there aren't any other questions, but if you think of any, as of today, Richie and I are still, oh wait, there is a question. Michael Carney, go ahead, Mike.
Hi, this is Mike. Can you hear me okay?
Yeah.
So, putting up that slide kind of got me thinking about, just, I guess we can look at it again just to see if this is a correct sort of way to think about intervals for, let's say, like an injury determination. So from what I remember here, so we only had that one group A and formation B was statistically significant from the rest of these. But if we, let's say, had like, our upper interval there during a, baseline for an injury assessment. And so, we could see that for that formulation A 0.50 that we have, let's say, 25% of that population over that interval. And let's say that formulation B 0.5%, 100% of that of that population, could we start thinking about building an injury sort of curve with those percentages and not have to run any sort of other statistics, since our intervals were built off of the distributions of those populations? Is that maybe a way? Because I guess for injury, we always I always think of percentages, over time, loss or over time. And so I was just kind of trying to conceptualize how to interpret something like this in that respect.
My 2 cents on that is this is sort of a proof of concept, but there's not enough data to really generate a good dose response curve. So maybe it's sort of step 1 of a 2 or 3 step process. And I had done a lot of work with this particular test chemical, brodifacoum, over many years, and it has a very, very steep dose response curve. If one looks at clotting time is fine, clotting time is fine, clotting time is fine, then a slight increase in concentration, and it really, clotting time is not fine, and the system is failing. And I don't know with 25% versus 100% respondents, just those 2 points, if you could make much of a decision. I think you would really need some more intermediate responses. For other test chemicals I've worked with, that other anticoagulants, we've actually been able to do that, where we get a more reasonable dose response curve, where at one dose level, 1 of 4 individuals responds, at a slightly higher level, 2 of 4 individuals respond, and then at an even higher level, all of the individuals respond. So I don't know if this is a great data set to really make that kind of decision. I hope that answered your question.
Yeah, sort of. I was kind of just looking at these data just in generally, but like, I guess if you were confident in that you characterized your, reference intervals correctly, maybe you could start making some assumptions of 1 or 2 individuals outside of that, or 100% of your individuals outside of that, to get a general sense of your, percent of response, I guess. So, well, thank you.
Yeah. So Mike, I would go back to John's comment about a weight of evidence approach, where this right now is a good weight of evidence. And then also, if you do good partial responses, I'll look at something like a dose response curve if you can do it. I know field sites, you might not always have like a nice gradient for the dose response, but I would personally think a dose response curve is oftentimes a stronger, well, definitely for predictive, it's much stronger. So, but thank you, Mike. To give people maybe a little bit longer, John posted some stuff in chat. Also, Chris Tong works for the USGS and now is a statistician for the Bureau, and he's trying to do more. There's a statistical community of practice across the Department of Interior. It's part of the Evidence Act, where they're trying to get more. Chris, do you want to do a quick ad for your group?
Thanks, Richie. Yeah, the Bureau statistician, that's me, newly hired. I have civil service tenure, so I didn't get canned the other week. My role here is to build statistical capacity at the U.S. Geological Survey, and that means helping people whose statistical expertise ranges from novice to advanced degree holder and everyone in between. So part of what I'm doing is accessing the training available to us in statistics at the Bureau, gaps in training, what can we add, what can we enhance, and coordinating with other people looking into training more broadly, cultivating a statistical community of practice to help, again, this whole range of constituencies, and adding resources, compiling resources. Oftentimes people in one part of the Bureau don't know what people in another part of the Bureau are doing. And so, for example, events like today's event could be of interest to lots of people. So I'm helping to promote these kinds of events within the Bureau, and I'll be meeting with Emily Silverman later this week to talk about coordinating with the Statistics and Evidence Community of Practice at the DOI level. So that's another thing I'll be pursuing. So thank you, Richie, for letting me do that.
You're welcome. Some people asked for the R package. I dropped it into the chat. So yeah, if there's no other questions, I can thank you for letting us present.
Thank you so much to our presenters. That concludes today's presentation. Have a great day, everyone.